Yeah, firstly, the sequencing results that we have to date have not been carried out in Gibraltar as such. We have been sending the viral RNA from samples from COVID patients in Gibraltar to Public Health England, where they have been undertaking the whole genome sequencing for us. And what it has shown is essentially that Gibraltar mirrors the progression of variants and strains that has been seen largely across most of Western Europe, and sort of mirrors that has been seen in the UK and in Spain. So we started out in the first half of 2020 with the original strain, and then over the course of the late summer and autumn, B.1 was replaced by a new strain, which has been labelled by some sectors of the press in the UK as the Spanish strain because it was first discovered in Spain, which is B.1.177. And that became our predominant strain throughout the autumn. But then towards December, January time, we began to see the emergence of one of the variants that has been reported in the press, the Kent variant or the UK variant, which is B.1.1.7. And that has sort of intermingled with the Spanish strain throughout our very intense third wave of COVID infection in December, January, and February of this year.
And is this information that you've gathered consistent with what we've seen across Western Europe?
It's certainly consistent with the pattern that we have seen in our practice in the NHS, where we have seen sequentially one strain take over from another strain. And in evolutionary terms, when new strains emerge and are successful, it's because they have a characteristic that makes them succeed over a previous strain, and often that may relate to increased transmissibility, and therefore that strain succeeds and supplants the previous strain that was in existence. And certainly what we've, from the data we have so far from our strains in Gibraltar, it follows what we are seeing in the UK.
And how can the science be applied to the vaccine itself, given that there is a suspicion that the vaccine may not be as effective against certain variants?
Yes, I mean, as I mentioned, the actual genome encodes a protein. And one of the proteins that is most relevant in SARS-2 is something called the spike protein. The spike protein sits on the outer surface of the virus and is what the virus uses to attach to our cells, to our mucosal surfaces, in order to cause infection and gain entry into our cells. The spike protein is also relevant because the vaccines that we have available to us at the minute, the Pfizer vaccine, the AstraZeneca vaccine, show our immune system a copy of that spike protein from one of the earlier viruses and teach our immune system to make an immune response to that particular spike protein. If there are variants that have mutations that change the shape of that protein, then there's the risk that the immunity that the vaccination delivers cannot bind to the new protein quite as well, and therefore the vaccine-derived immunity is not as comprehensive or as effective against new variants because the spike protein has changed in its shape. And that is certainly one of the concerns at the moment with some of the other variants. Not, I must add, the Kent variant. The Kent variant shows very good susceptibility to the antibodies that are generated by the Pfizer and AstraZeneca and other vaccines. But there are other variants of concern in existence, namely a South African variant and a variant from Brazil that are actually a little bit more concerning in that respect. None of those variants are very prevalent across Western Europe. They're not prevalent in the UK, only very small cases of those, and we have not as yet discovered any of those variants in any of the samples that have been sequenced on patients from Gibraltar.